In this video, we're going to learn how to extract DNA from a banana. We are going to need 3 cups, a knife, a tablespoon, and a teaspoon, a sifter, a funnel, and a transparent container. We're going to need a banana to extract the DNA. It's better if it's ripe. Then, we're going to need dishwashing liquid, water, and alcohol, as pure as you can find it. So here is the total of our materials. In addition, we're going to be using a chopping board. First, we're going to chop the banana. Here, we'll only be using 1/2 of a banana. A full banana should be enough for 5 to 10 samples. Make sure the banana is chopped very, very finely. Next, we're going to mash the banana. Make sure you end up with a very smooth consistency. We will need about 1 teaspoon of liquid soap and 5 tablespoons of water. This is very approximate, so don't worry if you don't get the exact proportions that we're saying here. DNA is trapped well inside the cells of the banana, so we're going to have to break it loose. The soap will help us do this by disassembling the membranes that are made of fats. We then need to mix very well so that we make sure that as much DNA as possible is released from the cells. If you have a sifter with a very thin mesh, you can use it to filter the mix. The DNA will be dissolved in the water that pours through, so now you know why we added the water in the lysis solution. This will take a few minutes, but after a while you should end up with a crystalline liquid or semi-crystalline, depending on how thin your mesh is. Carefully transfer some of the liquid into the transparent container. A couple of fingers in height will do. This liquid contains the DNA of the banana dissolved in it, so we want to precipitate it, which means to take it out of solution. For that, we're going to use alcohol. As it pours in, the water that is now dissolving the DNA is going to instead go with the alcohol, so the DNA has no option but to precipitate. That's banana DNA. It is not pure, it has RNA, it has proteins, other bits and pieces that got dissolved in the water, but it's mostly DNA. Now, let's try to fish some of it out. Take a toothpick and then try to swirl it and see if we can get any of the DNA out. Since DNA is such a long molecule, the DNA of one cell gets entangled with the DNA of the other cells, such that it's very easy to pull them out. Voila!